Hello dear students, welcome to the class of M.sc Zoology part 1. Okay, so myself uh, Hemant Asne, okay, I am working as a faculty to the KJ Somaiya College at Department of Zoology, KJ Somaiya College, Kopargaon. Okay, so I am here to discuss today with you people that with the that syllabus discussion for M.sc Zoology Part 1. Okay, and the syllabus which is for the colleges which are affiliated to the Savitribai Phule Pune University, Ganesh Khinda, Pune. Okay, so you have the choice based credit based system syllabus pattern, okay, which is revised in the year 2019, okay, and which to be implemented from the academic year 2019-20 to onwards. Okay, so syllabus pattern, the credit pattern will be discussed by one of our colleague to you people. So my job for today will be get touch with you people for being discussed for the MSc Zoology syllabus. Okay, and we are discussing on the course code named as the ZOUT 112, okay, which is the course code of the subject named as the cell biology and developmental biology. Okay, so the cell biology part will be taken by our HOD sir, okay, and the second part of this subject, that is developmental biology, is uh, going to be my side. Okay, so the syllabus discussion for the developmental biology, okay, we will get touched through the syllabus discussion, and the lectures is given to complete that particular topic. Okay, so as you know, the developmental biology, okay, so let get start with the syllabus discussion. Okay, the topic itself is giving you a hint about what the subject is you going to study. Okay, so the our subject topic will be the developmental biology. Okay, the developmental is nothing but the uh, various step by step processes to get an adult, okay, from an developing uh, stages of the one particular living organisms. Okay, that is nothing but the developmental biology. And what will be the definition for the developmental biology is, it is the branch of biology, simply it is a branch of biology that studies the processes, okay, by which the multicellular organism are grow and develop in the and controlled by their genes. Okay, and with the step by step processes which are going to get increase um, their circumstances in between their developmental stages. Okay, as you know, the development starts from the uh, formation of the zygote itself. Okay, so as you know, the egg which is come from a mother, okay, and the sperm which is come from the father, okay, and that after that fertilization, the zygote will be formed. Okay, at the point of that fertilization itself will the start of your developmental stages. Okay, the all the cells or the cell organelles are given by your egg which is come from your mother. Okay, but all your cell organelles are given from your mother. But what the sperm donation or the sperm composites into the egg, that only the set of chromosome. Okay, when that egg is simply non fertilized egg, that will be having the n number amount of the chromosome of set, that is 21 pairs. Okay, and that after that fertilization, the 2n number of the chromosomes are going to be in that particular zygote. Okay, but all other cytoplasmic structures are given by the mother cell. Okay, hence the degeneration of the cell organelles into the sperm itself, sperm can't contribute um, the cell organelles to the zygote. Okay, but all the cell organelles are given by means of the our mother cell. Okay, so the definition itself says it is the branch of biology, that studies um, the processes by which the multicellular organism will grow step by step. Okay, so it is a continuous process after the fertilization. Okay, so we will study the developmental biology. The first chapter in our syllabi is the introduction to the developmental biology. Okay, we are going to study the early theories, the postulate theories of the developmental biology are coined by the various greater thinkers, okay, of the biology. Okay, such as the we will see the Aristotle theory. Okay, we will see the various developmental theories. Okay, we will see the organogenesis theory. Okay, and we will see some concepts, that common concepts of developmental biology, like growth. Okay, so growth is a phenomenon. Okay, it is a continuous process, growth. Okay, and that progressive development of a living thing is nothing but the growth. Then the cell division, what will be the cell division is the bifurcation or the proliferation of a cell. Okay, is nothing but the cell division, and it happen by two ways, either the meiotic way or mitotic way. Okay, we will see what is they mean by the meiosis, what is mean by the mitosis. Okay, in these chapters. Okay, the general definitions, which are general concepts of the developmental biology. In that we will see the cell differentiation after the zygote formation, or the after the blastula formation into the developing embryo. Okay, so the cell get differentiated into the specific type of the cells. Okay, where that cell differentiation is occur and form the three different layer known as the ectoderm, mesoderm and the endoderm, whereas these three different layer are going to give the rise of the different systems. Okay, and that rise of that systems from the three germ layers is nothing but the cell differentiation, that is the origin of the cell differentiation. Cell communication will be the next terminology, and cell communication is nothing but the cell is communicate with the other neighboring cell of its neighbor. Okay, likewise, why what we are communicating with the one person to another, likewise cells are going to communicate with the signals. Okay, and that signals are going to get communicate within the cell themselves. Okay, and the cell communication process after by means of the cell signaling. Okay, we will see some cell signaling processes. Okay, by means of the cell signaling, the one cell is giving to the signal, or the that messages are get passed while the developmental stages are on the way. Okay, and at that time the patterning, okay, that pattern of the each differentiation cells we will see at the time of the patterning. Okay, what will be the induction and the competence will be the next terminology. Okay, the induction and the competence is nothing but the uh, in embryology, the process of one tissue going to influences the development of the other. Okay, so that will be the induction. Okay, and what will be the competence? Okay, the competence will be the this is the ability of the particular cells, okay, which is giving to respond to a specific inductive signals, okay, and that specific inductive signals is going to having some differences, some major differences, is nothing but the competence, that ability of the that selective cells to respond for the specific signals. Okay, and then we are going to study the second chapter, which is the model organism. Okay, what do you mean by the model organisms? Are there are the some models, okay, as like we are approaching towards the phenomena as a development. Okay, and at each developmental stages, there are some examples or there are some models which on which the experimental purposes, that models are get selected. Okay, and if their models having the some specific terminologies, the specific characteristics on that particular model of each classes, okay, at that time we can understand, for the better understanding for the human purposes, okay, why the humans are not used as a model organism, because there are some ethics. As you know, there are some ethics come in between to the human races. We can't use as human race as a model organism, but there is a differentiation into the gestation period in between these particular animals which are using for as a model into the studies. Okay, that various embryological studies are taken on these models. Okay, such as invertebrate model will be the Drosophila melanogaster. Okay, it is the one particular type of the insect. It's like a fruit fly. Okay, and which is has used into the various studies, like then it having the three type of the genes, that are bicoid, nanop, and the hunchback. Okay, so these particular processes as having with the this invertebrate name as the Drosophila melanogaster. Okay, so in fishes, the zebrafish, that is the Danio retorio. Okay, that Danio rerio is used as the model into the Pisces families. Okay, as an amphibians, the Xenophis levis is the frog species which is used as the model organism in from the amphibians. Okay, whereas the various developmental stages like the signaling, patterning, induction, and competence are seen into the this Xenopus levis. Okay, as a uh, bird, the model organism is used as a chicken. Okay, so chicken will be the best model organism to study the embryological derivatives, to have what the total procedure, okay, into the bird, as the it having the proper amount of yolk, and it having the uh, best way to get find to get touch with the developmental biology. Okay, as we are mammals, the humans, but human can't be take as the model organism, but in mammal the category, the mouse has taken the as a model organism into the studies. Okay, as mouse development or the direct birth, all these thing characteristics are matches towards the gestation, like gestation period of the um, humans. Okay, so these are model organisms having the short lifespan. Okay, the multiple the variations, okay, and all these characteristics of the each model organism are taken into the consideration, okay, and the lectures allotted for these second topic, that is model organisms, are about the weightage of the 3 lectures, okay. Then we will see the gametogenesis. Okay, simply the gametogenesis is nothing but the genesis is nothing but the formation, and gameto is nothing but the gamete formation. Okay, where there as two separate gametes are, we know that the female gamete and the uh, uh, female and the male gamete, where the male gametogenesis is well, sperm sperm is a male, characteristics of the male, okay, and that spermatogenesis is nothing but the sperm formation, okay. Why, how that sperm formations are happen into the Leydig cells of the testes, we will see, okay. We will see the structure of the sperm, okay, it having the head, neck and the tail, the specifications into the head, the acrosomal cap, the nucleus, then the degeneration of the various cytoplasmic structures, and that happens, and that total remodulation of the structure into the sperm, okay. The motor functioning into the sperm, the tail, we will see the regulation of the sperm motility, how that sperm motility is regulated, okay, we will see over there. In the oogenesis, as a female gamete formation, okay, the oogenesis is nothing but the formation of the female gamete, and whereas the structure of ovum, we will see the having with the structure of ovum, the nucleus, the cell organelles, the cytoplasm, okay, and the various specifications of the ovum. We will see the pre-vitellogenesis, okay, the yolk formation, okay, the vitellogenesis, yolk formation, and before the yolk formation, pre-vitellogenesis, what are the processes, okay. We will see the post-vitellogenesis, after the formation of the yolk, what are the changes happen into the that structure of the ovum. We will see the fourth chapter. Before that, the lectures allotted for the third chapter, that will be the gametogenesis, are of the 2 lectures, okay. Then we will shift towards the fourth chapter, that is nothing but the fertilization. At a particular stage after the uh, sexual act, there will be the movement of the sperm towards the egg, okay, and after the ovum get fertilized, that process, that concept of the fertilization, we will see the types of the fertilization, which going to highlight in the process, we will better understand the fertilization process, okay. The species specific sperm attraction, whereas the specific chemical signals are there into the egg itself, that are going to attract, or that are into that cell to cell communicate in between the medium of each particular species, and that particular species having the that attractants, that chemical signals are with the into that medium, okay, and that going to attract that species specific sperm attraction, we will see in that point, okay. So the recognition of egg and sperm, how that recognition of egg of each species to the same species, that means each species having the specific signals to get attached, or to get income across with the specific signals with the specific type of the cells of the each species, okay. So then we will see the acrosome reaction, okay, how that particular sperm is going to fuse with the egg, okay, and what is that acrosome cap is having the particular type of the protein that going to bind and going to dissolve the membrane of the egg, and that sperm is going to enter into the um, into the egg, and that egg will then get fertilized. Okay, then we will see the signal transduction, the signal transduction in between, to get know about the molecular strategy to avoid to the polyspermy in the fertilization. That will be the fast block and the slow block reactions. Okay, to get avoid with the polyspermy is nothing but the one egg is fertilized by means of the more than one sperm. Okay, so to avoid that situation into the fertilization process. Okay, so there is a specific interactions, that is nothing but the um, polyspermy. To avoid polyspermy, that will be the fast block interaction. And how that fast block interaction, that going to harden that outer shell of the egg. Okay, so that the all other sperms do not enter into the egg. Okay, we will see in the chapter 4. Okay, and the lectures allotted for the 4th chapter will be the 4 lectures. Okay, so fifth part or the 5th chapter will be the types of the eggs. Okay, which are the based on the amount of distribution of the yolk. Okay, there are various type of the species. Okay, and that species differentiation leads to the distribution among the eggs by the distribution of the yolk. Okay, we will see the process of the um, bird formation, bird new young one formation by the cleavage. We will see the types of the cleavages and the significances of the cleavages. Okay, these for this 5th chapter, types of the eggs, the lecture allotted are 2 lectures. Okay, so the 6th chapter will be the morphogenesis. Okay, whereas the blastulation we will see in the particular organism known as the amphioxus, the blastulation in the frog, blastulation in the chick, blastulation in the mid blastular transitions. What are the blastular phases? Okay, after the uh, fertilization, then the formation of the embryo, then the gastrula, blastula, and then the organogenesis. Okay, so the morphogenesis will be offered by means of at the time whereas the blastula phase will be ready. Okay, it will be shown. We will compare in between the amphioxus with the frog, frog with the chick. Okay, and then we will see the comparative study of the gastrulation or the neurulation in between the frog and the chick. Okay, the neurulation, formation of the nervous systems. Okay, we will see what are the situations in between the frog as well as the chick. We will see the organogenesis at the time the blastula is formed. Okay, where there will be the cells is going to differentiate into the various cells, then we can say that that will be the stage of the organogenesis. That means formation of the organs. Okay, and then that will be the developmental of the vertebrate eye, how that vertebrate eye is formed, formation of the eye field or the eyelids. Okay, then we will take the our point towards the cell differentiation, how cells are going to differentiate, formation of the ectoderm, mesoderm, endoderm, and then we will form how that various systems are going to develop into the developmental biology. Okay, and for the 6th chapter, that will be the morphogenesis, and the lectures allotted are 5 lectures. Okay, then we will see the pattern formation, this into the 7th lecture or the 7th chapter. Okay, then it will be the setting up with the body axis. Okay, there are the three type of the axis mainly. First will be the animal vegetal axis. We will see in the amphibians what are the animal vegetal axis is that animal pole and the vegetal pole into the amphibian egg. We will see the various developmental stages, how that animal pole is get decided by means of the when the sperm is going to fertilize at the animal pole section. Okay, and at that time that animal when there is a sperm is going to fuse with the egg, that part will be decided as the mouth or the ventral structure of the amphibians or the at uh, respective the various structures. Okay, and the at 180-degree, the at that point is decided as the anus of that particular organism. Okay, when that animal vegetal axis formation and dorsal ventral axis formation into the amphibians we will see at the time of the 7th chapter. Okay, then the anterior posterior axis formation into the Drosophila, the role of bicoid, nanos and hunchback gene. As I said, this will be the role of these particular three hormones. Okay, the bicoid, nanos and the hunchback, to how to get uh, form these anterior posterior axis and the development stages of the organs into the Drosophila. Okay, the lectures allotted for the 7th chapter will be the 4 lectures. Okay, then we will see the 8th chapter, that is nothing but the organizers. Okay, that organizers means the that specimen and the mangold, that primary embryonic inductions into the um, developmental stages. Okay, and that will be get organized to perform the functions or the organizers. And what are the functions of that organizers? That are nothing but the some part or the particles into the developmental stages while the studying of the embryology. And what are the part, the function of the organizers are molecular mechanism of the amphibian axis formation, how that molecular mechanism of the amphibian axis form. Okay, for the chapter 8 there are 2 lectures are allotted to complete this specific topic. Then the second last topic, that is nothing but the 9th chapter, is regeneration. Okay, that how the regeneration means regenerate. Okay, that means once we cut off that part, that part is going to regenerate over there where that part is going to get we will see regeneration in the salamander, we will see regeneration in the hydra. Okay, you might be know that each, if you chop off the hydra, that each cell of hydra can generate the uh, normal hydra from each cell. Okay, that is nothing but the regeneration process. Okay, that the regeneration process decides that when, if anything is cut off from one particular point, okay, at that point it going to get regenerate these all limb, its tail, okay, its body parts. Okay, so that will we see into the regeneration chapter. Lectures allotted for the regeneration are the 3 lectures. Then the last but not least will be the 10th chapter, is nothing but the apoptosis, aging and the senescence. Okay, the apoptosis is nothing but the program cell death. Okay, and that program cell death is having the importance into the formation of the embryo to the uh, adult. that okay, and that when the digit formations, the organ formations, the various specifications, the your intestine formation, your where there as a lumen present, where there as a lumen present, there is the importance of this apoptosis. Okay, the cell aging and the senescence, that at one particular time the particular cell is going to get age. That means the age of that particular cell, um, after the going to finish. Okay, and after the aging, the apoptosis is going to play an important role in your body to get shed off with the aged cells or the dead cell materials. Okay, and the senescence we will see into the 10th chapter, and the lectures allotted for this topic will be the 2 lectures. In total, these are the 30 lectures allotted for the developmental biology section into the course code ZOUT 112, that is nothing but the cell biology and the developmental biology. 30 lectures for the cell biology and the 30 lectures for the developmental biology. In total, this will possesses 4 credits. Okay, in that 2 credits for the cell biology and 2 credits for the developmental biology. Okay, so we will meet into the next session. Thank you. Okay.